Welcome back to Translating Proteomics. On today's episode, we wanted to give an introduction to proteomics for folks who are new to the field. We envision this episode as being a great foundation before diving into the other episodes.
Yeah, I'm looking forward to the session as well. And maybe just to start off, what is proteomics?
So, proteomics begins with the thesis that proteins are major drivers of biology and have impacts all the way from what happens inside each of your cells to large-scale physiologic processes and disease. And I often also try to explain that when people think about proteins, what immediately comes to mind is dietary protein on your cereal box or on your yogurt and says, I've got this much protein. But the reality is that there are tens of thousands of different proteins that are modified in so many different ways. And those proteins orchestrate a complex and beautiful dance that drives how biology happens. Proteomics is the study of that dance. What are the kinds of key questions that people are trying to use proteomics to answer?
So the four questions that people typically ask in proteomics is how much of the protein is in the cell? What might be different between two populations of cells? Is the protein modified? How is it modified? How does the protein interact with other proteins? And then ultimately, what is the structure and how does the structure change in a biological interactions? So why is it so critical to measure this myriad of changes in the proteome?
Ultimately, what we care about is we care about why a cell, why a tissue, why an organism um, performs as it does. We want to be able to build models of understanding that allow us to predict, based on some perturbation or some variation or some input, what's going to happen. So, for example, if I dump a particular chemical on a cell, how is that cell going to react? If the cell is in an area that's low in oxygen or has a lot of sugar, what's going to happen? How is it going to change? What occurs when transitioning from a normal healthy cell to a cancerous cell, a disease cell? And these kinds of questions that you alluded to are informed by what happens in the proteome. So, for example, let's focus in on just the question of transition from a healthy cell to cancer cell. What we might believe is that that occurs. There's a theory that says there are genetic mutations, and those genetic mutations cause differences in how the cell behaves. Well, what are those differences? Does the cell become squishier and more? Does it move faster? Does it grow faster? All of those processes that govern life are actually driven by proteins. And so the reason that we want to study proteins is so that we can understand what are those processes, how those processes are changed, and to what extent are the proteins the drivers of those processes. The genome captures very well with that there are alterations. The transcriptome is sort of an intermediate step, but the proteins are actually doing the work.
So what kind of things can you do with proteomics and what can you not do with proteomics?
When one thinks about what is an easy thing to do in proteomics versus a hard thing to do, there are really two axes to think about. The first is how complicated is the sample that we're looking at? Is it a single protein? Is it a large number of proteins? There's also a question of just how many samples are we looking at? Are we looking at 1, 100, 1,000, 30,000? And so as we move that slider from studying an individual protein all the way to studying, whole population studies, things get harder and harder and harder. And really where proteomics continues to struggle and is just now in the last few years making tremendous strides is on that population scale proteomics. We've seen a number of studies recently, we've seen a number of studies announced that are on the scale of tens of thousands of patients. Andreas, one of the key drivers of what we can do in proteomics has historically been the tools and methods and techniques that are available. When you think about the landscape and how it's evolved and how frankly, you were a driver of that evolution. What are the classes and canons and of methods that are available to us in proteomics.
So in terms of technologies, one can probably put them in certain categories on how you address particular questions that you might have about the proteome. So typically, protein microarrays, mass spectrometry would give you the ability to measure what's the difference between two samples and study the composition of samples. 2D gels would also fall in a category, probably not as popular anymore. But when you want to then understand more about, for example, protein interactions, you would have to go and use Q purification mass spectrometry approaches. And obviously, as you're trying to understand protein function or structure, you would use more things like X-ray crystallography, NMR spectroscopy, or even cryo-electron microscopy. So for each of those questions you ask from the proteome, there is a set of tools that we can use today. What do I have to pay attention to when we're trying to understand protein or the proteomes? What are the pitfalls?
I think the first thing that one needs to think about, and this is invariant of the technique that you use, is where did the sample come from? And what are all of the pre-analytical variables that influence the quality and fidelity of that sample? Some of those even precede that and have to do with study design. So when we're looking at temporal processes, we need to know, are we even measuring at the right time point? Or are we looking at a time point where the change we're looking for hasn't happened yet or is long since over? For example, if I'm really looking for signaling cascades that change phosphorylation, some of those changes can happen in the second to minute timescale. And so starting from a study design that is aware of where and when the effect I'm looking for is likely to occur can be critical in actually seeing it. Beyond how the sample got into its current state, there are also different challenges associated with how you prepare the samples. So maybe can you tell us a little bit about what are the different things one needs to be aware of in the sample preparation for proteomics analysis?
Certainly happy to do so. I think my experience tells me that if things went wrong in proteomics in my past, it was often the sample preparation that went wrong and then really made it impossible to get a good readout, regardless of what techniques you used. And so happy to talk about it. I think some of the sample preparation challenges are sort of similar to what you mentioned in the pre-analytical variables, right? Protein degradation. Proteins can still degrade during the sample preparation process. Obviously, during sample preparation, you can also cause contamination. You certainly can lose protein material. And then you might not be able to efficiently recover the proteins that you are intending to isolate. I can only recommend if you enter in this field that you talk to someone with experience in this area, and then discuss it with the people that will run these experiments to make sure that you cover your bases when it comes to, do we recover all the proteins? Have we actually extracted those proteins? And once the proteins are extracted, make sure that we don't lose them through degradation or loss. And then ultimately make sure we don't contaminate the sample by using the wrong or incorrect labware.
One of the other really significant challenges that we face in proteomics is, of course, on the data analysis side. And the challenges there stem from, first, how do we even think about temporal patterns and whether a protein is going up or going down or chaotically behaved? Did we sample sufficiently across the timing? Did we have timing at all? And the canon of tools for doing binary differential expression is very well established. The canon of tools for looking for trends that vary over time is still pretty new. So that's one of our largest challenges, is that there's just not a huge standard body of work to draw on. One of the other challenges that we have, particularly in shotgun or bottom mass spectrometry, is that we analyze peptides and then we have to infer the protein that was present and its abundance from the set of peptides. Unfortunately, not all peptides are unique to every protein. Some proteins may be identified with one or two or 20 different peptides. And so the integration of peptide information into protein abundance is actually an incredibly non-trivial challenge. I'd say the last challenge we really face is in multi-omic integration with proteomics. Again, it's such a bleeding edge area that there's no standard toolkit to draw on. And factors like how does the transcript or how does the gene lead to mutations? And did we capture that when we were searching for the protein in the proteome? And then how do the transcript abundances relate to the protein abundances? Those are all really sophisticated questions that we just don't have a good toolbox for as yet. So far, Andreas, we've talked a lot about proteomics as it is today, and what are the details of individual techniques and the canon of what is being done extensively throughout the field. But there are a number of really exciting new areas that are expanding, and so can you touch on a little bit what is emerging? What are people excited about in proteomics?
Absolutely. Yeah, there's a number of techniques that are sort of like better versions of what we do today that address the diversity of proteins, but also the spatial and temporal aspects and the functional aspects of proteins, right? So let me mention the spatial proteomics. That's a rapidly evolving field. I think the original technology was really multi-imaging, where people literally take an image of a tissue using a laser ablation approach. That approach is now, being expanded using proximity labeling, where you use techniques like BioID or APEX to really map proteins at specific neighborhoods, whether they're organelles or cellular structures. And then there's obviously the evolving spatial proteomics techniques that use affinity-based reagent to really understand how are proteins localized within the tissue. So maybe another approach that is getting more and more attention is the field of chemoproteomics. In this particular case, you combine the chemical biology with some of the core technologies we described above, whether that's to actually measure the activity of proteins, or you measure the actual binding of proteins to other proteins, or for particular to drugs. So specific approaches are being developed and are used now to really understand, when I put a drug into a cell, where does it end up? Where is it binding to? And that really helps in understanding not only where the spatial context is, but what kind of action does the particular drug molecule have in a particular proteomic environment. The other area I should mention here is what is now known as single-cell proteomics. That technique is rather new, been around for a few years, but technology is rapidly being developed to really observe what is in a single cell and how does it change if I look across thousands of cells. Lastly, I would mention another field that's very, very early, but ultimately will emerge in the next few years, which is single molecule proteomics. So in this case, interested in studying, every single molecule you might have in a particular sample, and understand the heterogeneity of that sample in much more detail than it's possible with the current technologies.
Thanks, Andreas. It sounds like the future of proteomics is bright, and I'm excited to see what's going to be emerging over the next decade. Thanks again for joining us today on Translating Proteomics. We hope that this overview of the field has really helped you build a foundation to dive deeper into this really important area of science. If you have other questions or thoughts, hit us up in the comments or shoot us an email. Thanks again for joining us today.
We hope you enjoyed the Translating Proteomics podcast, brought to you by Nautilus Biotechnology. To contact us or for further information, please email translatingproteomics@nautilus.bio or scan the QR code on your screen.